I think people are probably joining. So welcome everyone to the next session in RBQM Live. We hope that all of you who just attended our last session regarding the ICH guidance updates found the insights that Jonathan shared extremely valuable. Thank you for joining our next session of the day, which is a panel discussion around interpreting QTLs through the lens of where they are today and their future evolution. My name is Steve Young. I'm the Chief Scientific Officer at CluePoints, and I'll be moderating our discussion today. I am honored to introduce our panelists for this discussion. First, I'd like to introduce Steve Gilbert, the Senior Director of Statistics at Pfizer. He has worked in clinical trials since the late 1990s, first in CROs and then in pharma, supporting all phases of drug development in numerous therapeutic areas. Steve is a member of the Statistical Research and Innovation Group, which provides statistical consulting and technical support across Pfizer. Steve's consulting includes risk-based monitoring, clinical statistics, and quality research. Welcome, Steve.
Thank you.
Next, I'd like to introduce Joanna Florek-Marwitz, the Head of Risk Management and Data Quality at UCB. Joanna has extensive experience in clinical and non-interventional trials operations across all therapeutic areas and is an expert in applying program management, integrated risk-based quality management, process improvement, innovation, and change management methodologies. Welcome, Joanna. And last but not least, hi. And last but certainly not least, I'd like to introduce our third panelist, Marion Wolfs, a senior leader within the Integrated Data Analytics and Reporting organization from Janssen. In this role, she is responsible for the oversight, execution, and management of the analytical risk-based monitoring processes, including beyond trial level risk analysis at Janssen. Welcome, Marion. And just an initial disclaimer on behalf of all of the panelists, and for avoidance of doubt, they are all speaking for themselves today and not for their organizations. Before we get the conversation started, I wanted to take a moment to walk through the console for our session today to help you better navigate what you see on your screen for a great experience throughout this session and all of RBQM Live 2022. On your screen, you'll see a window for slides, a window for video, some more highlights on our speakers, and a Q&A box where we encourage all of you to submit questions throughout the session that we'll take some time to answer at the end. All of these windows can be adjusted to your preferred viewing size for an optimal event experience. You will also see a Take Action tool that will remind you to click at the end of this session to take you directly to the next session of today's event. So now let's get started. So to start off the panel discussion, just really a simple question for our panelists. What do we mean by the term QTL, or quality tolerance limit? So really a basic, just contextual question. And do you see any challenges in establishing a common understanding so far with regard to that? And Steve Gilbert, maybe you can start with that one. What are your thoughts?
Yeah, sure. Thanks, Steve. The funny thing was that QTLs are one of the first things that I had worked on with our central monitors and basically risk-based monitoring in general within Pfizer. And so when I first heard about it from a mathematical background, quality tolerance limit is one number. And I'm like, what's all the problems finding one number? And then as I became exposed to it, I found out that, yeah, QTL is used to mean many overlapping things, including this entire process where we go from critical to quality factors to identifying risks to something to measure to actually measuring it and everything we're doing. So at this point, when I still talk to study teams that haven't been exposed and it's new to them, which we have fewer and fewer as time goes on, break it up into pieces. There is the entire QTL process, which is defining it and measuring it and looking at things at study meetings, and then have to add a little extra nomenclature so we don't confuse ourselves. So once we identify a risk and decide how it's going to be measured, like percentage of people not lost to follow-up, then we have a parameter. So I'm careful to say parameter. And then when we talk about the actual, what I would call the quality tolerance limit, the threshold where we're possibly could have problems with safety or the validity of our results. And big one that gets everybody, actually that's probably the toughest one, is finding the parameter. And then also, what do they expect it to look like if everything is going well? So the expected value of it. So I add in a little extra verbiage to QTL to hopefully keep from confusing ourselves with it. So.
Yeah, that makes sense. So what you're breaking it up into two components. There's the QTL parameter, which is the actual thing you're measuring, and then the actual limit value or the threshold value as the other piece of it, right?
There's the other piece of it, and then of course the whole regulatory part where we have to keep track of it and look at it, and that's also, some people just
the whole
say
process.
QTL.
The whole process around it. Yeah, no, very interesting. Thank you. So Joanna, anything, does that make sense to you? Anything to add to what Steve said there?
Yeah, that's a very comprehensive definition already, so it's hard to add, but I would like to emphasize the challenges related to the understanding. Why do we need basically a QTL, and how to establish the QTL, on the study level? What is the study level specific value, and what is the general purpose of setting up the QTL on the study level? Because we have been for many decades in the clinical research without the QTL. So now, to make the people understand that we are just focusing to identify systemic issues on the study level. This was something, let's say, from the change management perspective was and is still very interesting aspect, I think.
Very interesting, thank you. And Marion, anything to add?
Yes, absolutely, Steve. So I think to the first question, right, what do you mean by QTL? What does it mean for our studies at Janssen? And I think similar to Joanna's and Steve's explanation, we really tailor the whole quality tolerance limits process to the scientific questions of a study. And I think the confusion, maybe if you talk about the challenging in establishing a common understanding, is related where it originates from, right? So the whole QTL word, if you will, comes from the manufacturing piece, where it's very clear cut. What does it mean? Clinical trials obviously are not. So you can put thresholds on almost anything, KPIs, KRIs. So there are absolutely different ways how to make the interpretation. So I think that's the challenge that we see currently within the industry.
No, thank you. Yeah, that makes sense. Certainly rings true with me as well, in my experience. And I guess maybe we'll get into some of those challenges a little deeper on as the panel discussion continues today. But thank you. So
Steve,
let me get
still
to
have 1 minute. I would like to add that the QTL, which is really should not be understood as self-fulfilling prophecy, right? Because we will see no causality very often between the study quality, the trial quality, or the outcome of the data, and the QTLs being set up or breached or managed on the study level. So really coming back to the understanding of the challenges, this is something that we need to be very transparent with the study team to explain, like what is the purpose and what is the expectation, so there is no doubt.
Yeah, makes perfect sense. Thank you, Joanna. So, a follow-on question, besides GCP compliance, which I'm sure is, always is definitely a motivation with the introduction of QTLs in E6R2. But what besides that do we see, or do you see as the primary motivations for implementing QTLs? In other words, what are the primary value propositions for QTLs as you see it? Joanna, do you want to start with that one?
Yeah, sure. Well, obviously, you mentioned already there is a regulatory requirement But I would like to emphasize the ethical motivation as one of the primary motivations to implement the QTLs. And basically, if you think about the process and the definitions and the people involved, this is not really much different to the past, right? It's where we have seen studies with no QTLs, and we still have studies with no QTLs. But the overall, the aim of the primary motivation to implement them really should be at any stage the our conducting study safely and in reliable research, and providing reliable research really for patients, because we could be patient overnight and become patient overnight. So if we think about this ethical motivation, I think this gives us additional perspective to the implementation of the QTLs and the reason why, or the motivation why we do this. I think we should not use the QTLs also to showcase the better quality on our studies. So, and there is a temptation, like if you set up the QTLs, you think the study is, this is the confirmation for the study quality and reliability and safety of the patient. So we should really resist this temptation to, and between organizations or within our organizations, to produce or have the perception to produce better studies. From my perspective, I can add that I have seen successes in the QTLs being set up on a very high levels, and we are going to speak about the way how to set up on the study level what are the types of QTLs a little bit later. But process-wise, I think that to use the QTLs as part of a sponsor data surveillance and oversight of data surveillance being done by the CRO could be a very good motivation as well.
Great. That's all makes a lot of sense. Marion, what are your thoughts on motivations? Key value?
I fully agree with Joanna about the ethical component to it, right? We are in a business of drug development, medical solutions for patients, and we want to make sure that we do that in a safe as possible way, that we develop medications that are as safe as possible, that they do what they intended to do. And with that, we need to make sure that the clinical trials that we do, that really are scientific projects, if you will, that they are conducted as intended, and at the end, we are able to answer these scientific questions in the right way. And with that, I also see the added value there here, that the whole process makes you look into that scientific question deeper. Everyone involved in the study team now has to really grasp and understand what it is and what are the assumptions that we use when we do that. We think up front, and how is that going to evolve over time? Did we make a mistake in the assumptions? Is there anything that came up that we did not have control on, and so on. So that's really where I see the added values of quality tolerance limits.
Yep, excellent. Thank you, Marion. Steve, anything to add in terms of value prop?
No, well, I totally agree with everything that's been said. I also agree, yeah, it keeps the study teams focused on what's important. It's been a value at Pfizer and during studies to make sure that we were getting labs in when we were supposed to and things like that, that maybe they would have looked at otherwise, but once it was raised to a level of a QTL, it had the attention of an entire study team, and it was done in a timely manner. And
yep.
I can't agree more with the ethics of it. It's not just research. We're not just finding drugs. We do this in partnership with our patients, and we're responsible for keeping them safe, and we're responsible for making sure that their commitment to our trial is we're not wasting any data. We haven't run a study that we can't get a clean readout on because of quality issues. That's not ethical or fair to any of our patients.
Yeah, no, makes perfect sense. So it looks like very strong agreement on the ethical motivation around QTLs. And it's something I've also observed before, when thinking about QTLs is, from a quality oversight perspective during trials, what can be more important than monitoring for important systemic level risks or issues on your trial, right? So which I think is what the spirit of QTLs really represents as well, correct? So very good, thank you all for that. So next question, what do you see as the scope of QTL implementation, either currently what you see and maybe what it should be? For example, do you see certain studies that QTLs might not be applicable for within a portfolio of studies, or what are your thoughts on that? And Marion, maybe I'll start with you on this one.
Thanks, Steve. So currently at Janssen, we implement QTLs from the early development trials within the interventional trials in medical affairs, so quite broadly. But we do have exceptions. So in our risk-based monitoring model, we apply different levels of central monitoring, if you will. So we call that the full support, compact support, and light support. Apologies, think about that a little bit. And for the light support, we don't implement QTLs. An example from a light support study would be a study with very limited number of patients, where that everyone that's working on the study team knows all the data inside out. So that would be a study for us where we currently do not implement quality tolerance limits. It helps us to focus the implementation on the most critical studies, I think. It also if I say easy, that's not the right wording, but it's less difficult to do that for a little bit larger studies in the late development and even within the medical affairs organization. We have seen benefit, for example, from implementing QTLs for ongoing studies first. So where you have already some data at your fingertips, specifically if you then want to set a threshold, you have something to start from to see if the methodology that you put in place for setting the threshold, does it make sense? Are we completely off from what we see? It's easier to implement QTLs in a larger program. You have different similar studies following each other, so you can build on what you already have. You have more historical data that you can use for threshold setting. All of that is so much more complex if you go to a very small early development study where you literally, you have nothing to start with, and things change so rapidly. So that's a little bit how we stand in that space. We still have challenges, I would say, with regards to the threshold setting. We are trying different things to really open up our opportunities to find out what the best approach is. We are currently now at the time that we are going to kind of bring back all of these different solutions and see what works best in which situation. Can we learn from that? And I really see this as a continuous learning environment for us internally, but I foresee for the broader industry as well.
Yeah, very interesting. Makes sense. Thanks, Marion. Steve, how about what's your perspective on scope?
Very similar to Marion's. We don't include very small studies, particularly early phase 1. You have patients essentially in-house, very small numbers that go in small groups. And I mean, we're constantly surveilling them for, any signs of safety issues. QTL can't really apply it very well there. And also, since we're, Pfizer, large company, we do many types of research that really aren't interventional. So they obviously don't fall under there, like quality improvement research and things like that. But we apply it as much as possible everywhere we can.
Yeah, so I've certainly heard that before as well about like phase 1 studies, and I guess like you say, because you're looking at everything, or the study team is looking at everything closely in those cases anyway and scrutinizing it. And I guess is it also because those studies tend to be extremely short duration?
They're short duration and so small numbers. Taking kind of a statistical, we're going to look at a parameter approach doesn't make that much sense. And when there is even greater concern than usual, I mean, there are other things that are put in, like if you're looking at small cohorts of maybe 6 patients at a time, many times if they're really worried about it, first we do a sentinel group with 2, and then we do the rest of, I mean, lots of very well-worn, trusted methods to minimize risk. Right. They're applied constantly.
Right. Yep, makes sense. Thank you, Steve. And Joanna, how about you?
I think our situation is a little bit different. I think our colleagues here, Steve and Marian, mentioned 2 processes which are really, centralized monitoring processes really set up internally. If you use the QTLs as a part of the sponsor data surveillance oversight, there is a different approach to this, and really study, where I fully agree with, is to have a study specific approach, if you think about centralized monitoring and QTLs as well. So first, we have been focusing on phase 2-3 studies. Secondly, for the centralized monitoring, also phase 1 study and post-approval safety and efficacy studies have been implemented. We don't consider this being done on the NIS studies at all. And as I mentioned, the phase 1 study is not phase 1 study. There are also some variations and definitions on the study needs may also vary. So we think that the best approach to this is to be really study specific and flexible, which is also combined with a particular level of standardization of the QTLs, and really to make it very clear and very transparent. Doesn't matter what type of study it is, that we would like to recognize issues early and prevent and mitigate, and focus on what's essential to patients. So change the QTLs are always very successful and very supportive on changing from to the prospective and preventive wave of actions.
Yep. No, thank you. That makes sense. Thank you, Joanna. So, and part of what your answer might already be addressing the next question that I have for the panel, which is around, kind of describing it. Can you summarize at a high level, what are your thoughts on the process for selecting QTLs for each study, in terms of maybe when it should be done, who should be doing it, and any other tidbits that you would suggest in terms of the selection process for QTLs? And Steve, can you start with that one?
Yeah, sure. Happy to. Again, we're still working this into, still not completely business as usual yet, but our goal is to work. One, it's the study team is going to choose the QTLs, and we make this part of the original assessment of risks. So they're in our so-called whiteboarding session, where they get together and find all the risks for the study and rate them and rank them. From that, we try to generate what's going to become study-specific KRIs, which I know we haven't mentioned yet or defined, and the QTLs that we'll use usually come out of that. Then we do have a library. We keep track across the company what we've done. It's not a pick list, use this, that one, but they're examples of what have been done before. And then within individual therapeutic areas, everybody, information is shared, and people know that last year we did a very similar study. This is what they did in the first place. You'll look, does that match with your current risks? Does that match with your design. You could reuse something that had been done by an earlier group, or just update it with new information that we might have.
Yeah. Interesting. No, very, very good. Thanks, Steve. So it really is, it's starting with, and it's connected with the risk assessment for your organization, with a pre-study.
Yeah, it's assessing with the risk. And again, and if you really want to go back to where it all starts, critical to quality factors should have gone before that, and, all the way up the list. So this is one small part of that. Makes sense.
So Joanna, is that similar to what your experience is with this and observations?
That's a very good way to address this, the selection of QTLs for on the study level. I can report, a little bit or add to this on a different approach where the QTLs, at the start, it was really, compromising between the adding the scientific value by implementing the QTLs on the study level and the feasibility of implementation, because it should not become, overburden, right, for the study team and for risk management team as well. So for this reason, in terms of implementation, we focus on the standard QTL parameters. We have a very short library, like currently 6 QTLs. On the study level, most probably you will see only like 3, maximum 4 QTLs being selected. Basically the challenges we have also seen, adding to what Steve already mentioned, was the taking the connections between the KRIs and QTLs. And this is very specific to the process being in place, and very specific to the fact that the QTLs are being used as data oversight, data surveillance oversight. So this the challenges are related more to the the implementation of the process itself and having this connection in this process. So we are also considering bringing those 2 steps together again, the KRIs and QTLs, to be really more efficient and improve our way of oversight.
Yeah, very interesting, Joanna. Thank you. That's and Marion, want to get your thoughts as well, and maybe you can also comment about I think both Joanna and Steve touched on KRIs connected with QTLs, and wondering your thoughts and as well in terms of what do you see as the relationship between those. But please add any thoughts about what do you think is the focus in selecting QTLs?
Absolutely, and again, right, it boils back to what is your definition of quality tolerance limits, right? You can have thresholds to a lot of things, to KRIs, to KPIs, and all of that adds value. It gives you a sense of where you are, when to take action. For us at Janssen, the QTL definition, again, right, is really connected to the scientific question of the study, because at the end, right, the data that you collect for the study will determine the outcome of your analysis, and then, whether or not you're successful in what you're trying to achieve. And although, for example, your completion of data in the TMF or timely enter of data is absolutely an important quality component, it's less directly related to, are you able at the end to answer these scientific questions for the study as well as patient safety related. So we also use a library, I think similar to the others. Although I must say, right, if you use a standard library, it takes a little bit away from the making it study specific. So what we do is the label is more generic. So for example, an evaluable endpoint or missed endpoints, but what that exactly looks like how you would define that for a specific study, you will then need to look into the detail. So we would typically have ballpark, I think, similar to Joanne, 2, 3, approximately. Most certainly not more than 5 is what I've seen so far, QTLs for a study. But we do have additional KRIs, right, for the more operational monitoring of things. I hope that answers.
KRIs are often, KRIs are often thought to be like for monitoring site level risk, or that's a perception out there.
No, you can roll up, right? You can roll up from subject site, even beyond the trial level. These KRIs have absolutely a lot of added value to identify systemic issue. Yeah.
That makes
Steve,
a lot of sense. So, yeah, go ahead, Steve.
I'd like to add to that. Yeah. Because we tend to stay a very few QTLs. It depends upon the study. Many studies have one or two. Our oncology studies, because they're it's called one study, but there are many parts to it, can have 5 or 6. So they have QTLs covering expansion versus dose finding. But as far as the scientific question, again, I hate to drop jargon on people they didn't want to hear, but we also have many of the QTLs we have revolve around being able to evaluate the primary endpoint, the scientific clinical question of the study. And for that, we have to go back to the estimand framework of how we're handling things. So just losing patients is not great, but depending upon exactly what we're trying to estimate and how we're going to do it, if we know why they left the study, it could still be informative and doesn't harm us on our primary analysis, whereas they just lost a follow-up, it may. Or if we're analyzing the study in a different way, even if we know why they left, it could still harm us. So you have to dig down a little bit into what's the goal of the study and how things are going to be, the statistical analysis at the end. And another part, one of the reasons we've leaned towards a smaller number of KRIs is if I link things to the estimand, to analyzing my primary endpoint for the study, what I might do is make some sort of composite of loss to follow-up and missing a lab. For example, for a vaccine study, I may need 2 labs. And I don't care if we lost the lab, if they left the study, no matter what it is, it hurts us. We don't have that lab value. But if I were to break that up into a standard loss to follow-up, missing separate KRIs, none of those single ones might actually breach a limit and alert me that there's a problem. But when I combine them all together, because they're all used for one purpose, then that limit is, I think, is more highly related to what I need to assess the risk of that study, and I'm less likely to miss something.
That's really interesting. But I also guess that the calculation, the aggregation of that combined sort of QTL might be more challenging and complex to implement. No, or is it?
Yes, it could. Yeah, it could be. But we should, we should be keeping track of disposition. We should be keeping tracks of labs throughout the study. So
you need to think about that anyway, Steve, right? Because it's complex. But if you want to do the analysis, you need to think about it. Even though it is complex.
Sense. Yeah, makes perfect sense. Very interesting. Yep.
And what is sometimes very helpful is really to select to think about the study specific thresholds for the QTLs, right? Is making this tailoring this really for the study needs and getting to the point of answering the scientific questions like Maria mentioned.
Yeah. So it sounds like there's definitely a common theme of, first of all, focusing on the scientific question or endpoints or estimands, as Steve said, and that sounds like a common theme or trend in terms of focusing on QTLs. So, and speaking of thresholds, Joanna, I'd like to ask that question. I mean, what are your thoughts on the appropriate process for selecting QTL thresholds? And for example, is it purely based on an assessment of available historical data? Are you using that, or are you getting it internally or from external references? or is it the statistical design of the study is driving the setting of thresholds, or is it a combination of both? But anyway, Joanna, why don't you take that one first?
Yeah, sure. So yeah, basically that's like you mentioned. This is a combination of all those aspects you already mentioned. So I can speak to the way how this is implemented based on historical data, where there is a particular amount of internal logged studies which has been put through the SAS program to generate basically a library of historical data and support the identification of QTL parameters on the study, applicable on the particular study, and supporting the identification of respective limits to this particular QTLs. So this is all being loaded in the visualization tool, and where you can basically select, related to therapeutic area, to compound, to study type, like double-blind study, open-label study. You can select a study phase and then really, try to make the best possible narrowed information, and then speak to your experts. So, for example, if you have to set up a QTL for inclusion-exclusion criteria not met, so you're looking at the proportion of a patient's randomized, or if you don't have randomization, then the patient dosed, with violation of any eligibility criteria, and then this proportion to all randomized patients, and then your statistician is going to support you, like Steve already mentioned, and he's driving this process. We have statisticians being involved and supporting our efforts to establish the QTLs, or to identify the QTLs and establish the QTL thresholds from the methodology perspective, from the modeling, from analytics. And if we come back to the inclusion-exclusion criteria not met, and we think like, okay, statistician will define, the number of patients in the per protocol analysis, and how this relates to this particular QTL, and how this needs to be considered in setting up this particular QTL, because you will see also eligibility evaluations not being the only reason to exclude patients from the per protocol analysis. So this needs to be considered, the historical data, and then the narrowing the results to the level where we work together with a statistician and see, how this methodology is being supported. So for this particular one, there were 4 historical studies, and with the highest inclusion-exclusion numbers, and then we, the particular limit. In this case, upper limit has been set up for the studies and considered as this for this QTL across the studies. So, in summary, there is historical data, very, very useful. I can recommend to everyone to take this effort. It's a particular work, a particular effort you need to put in, but it's very useful to have this opportunity to reflect to historical data internally. Then you have the stats methods and methodology. Then you have also the expert knowledge, like from medical experts, from stats as well. So basically, this is, from my perspective, a very good approach to go, with also at the same stage with some limitations, because if you have particular limitations in access to historical data, or to the to this expertise from medical experts and stats, this could also become a challenge. So at this stage, I would like to emphasize the QTLs and QTL setting up the QTL thresholds is something which is really a cross-functional and expert-driven process.
Very fascinating. Thank you so much,
Can I add maybe? Oh, sorry.
Please do, Marianne. Go ahead.
Yeah, no, I just want to add a little bit on the use of historical data piece, right? I fully agree with Joanna. It can be very helpful. It gives you kind of the first insight in what can it look like approximately. I do also see that there are limitations, right? You need to realize that assumptions that are being used for a new study, even if it's the same compound, the same patient population, because the question is different, the assumptions are different, so therefore that threshold can be very different from what you see in your historical data. Then also things change over time. So what is still an appropriate historical data to use to set a threshold in the now and in the time to come that the study will run, right? If there are big changes in how data has been collected, then maybe that looks different. So again, it all boils down to what do you assume? What are your assumptions and the estimates around what is the statistical setup of a clinical trial that you need to take very much into account, and then you may or may not overlay that with historical data that you have to do a little bit of a reflection of what that looks like. Now, what I've seen very much added value for our clinical trials is run scenarios. So you take your statistical setup and then you just try out, what if we would miss this or that? You just have them run different scenarios and then calculate the impact. That is seen very much added value as well.
Very interesting. Very
I
interesting.
agree on the historical data limitations for sure. But as I mentioned, expertise is the main to approach this.
Yeah, I'd like to add that.
So general theme here sounds like definitely very worthwhile to reference historical data, but there is some caution as well. You need to be careful in applying it to a prospective study. Steve, just in the interest of time, maybe just add, I'll give you about one or 2 minutes to add your thoughts.
Okay, yeah. I'd like to break that down a little bit more, kind of expanding on what Marion was speaking about, using the historical data. It's tough enough getting from teams what they expect to see as single number. I'll lose 15% of my patients. From the historical data, we can also kind of get variability across similar trials if we can identify similar trials, if we have. And that helps in secondary statistical limits, which we can use as an early warning to whether we're going to have a quality confidence limit threshold. And going back to the kind of process control, sometimes the QTL what I call the threshold, isn't really based on a statistical analysis. It's based on, I know in this therapeutic area, the FDA will not accept these results if I lose more than 20% of my patients, like if it's a pain or arthritis trial. If it's a depression trial, then they expect a much higher rate, for different areas. Sometimes those thresholds are based more on practice and really long-standing historical kind of data that's baked into the community. And an important thing that we can do with that is see whether or not that variability is putting it at risk of crossing that bigger threshold, or as capability analysis from what they do in quality control or in labs. And I think that's an important value add as we go into it, going, we're designing a study that's too small to give us high probability of actually meeting our goals. And we know we have to go back and change something from the beginning. It's not just, sometimes we have to go back to the drawing board.
Yeah. Makes a lot of sense. Thanks, Steve. So I'm going to give you each just 1 minute tops for this last question, because we do want to reserve some time to field some of the questions, and there's a lot of questions coming in, which is fantastic. But just real quickly for each of you, and I'll start with Marion on this one, how do you see implementation of QTLs evolving over time? Are there any key things that you think we should be anticipating in the coming year and beyond with QTLs?
I missed you a little bit, but I think I heard enough to answer the question, Steve. So it's about, what's in it, QTL-wise into the future. What's the future look of things?
Future, yes.
Is into the future. Yeah, in the future of things. I think it's an interesting area where we continue to learn how to do that. So it will add more value while we learn, become better in choosing the right parameters, setting added value thresholds. That's how I see kind of the future of quality tolerance limits.
Excellent, thank you. Joanna, how about you?
I fully agree with Mariam, but I would like to add that I see the QTLs expanding to the clinical data management area. I think we have, great. We have established this in the risk-based monitoring, and we have all this experience. And to translate this to the other areas like clinical data management would be a great opportunity, and really changing the way of thinking about, instead of fixing issues, preventing issues and mitigating risks in this area where there is a huge potential. I think that would be great.
Excellent. Thank you. And Steve?
What I'm hoping for the future is this becomes more business as usual and automated. When the study teams get together, we have the historical information can be pulled up easily so they can find those limits, so they can find those expected values, that it automatically we're starting now to incorporate. If we have a QTL, we have a corresponding KRI. And again, just kind of make it one seamless process.
Yeah, makes a lot of sense. Ken Getz gave a great keynote presentation, and one of the metrics he was showing was adoption of various components of RBQM, and I believe he had one for QTLs, and it was showing, well, this was probably a year old, but it was only 11% of organizations were implementing QTLs so far. So I assume we should all expect that we're probably hitting an inflection point, right? And that's going to go up dramatically as we move forward. So I want to move now to answering some of see if we can answer at least a couple of the questions. There's been a tremendous amount of great questions that are being entered by the audience during this session. There's so many questions, we're not going to have a chance, obviously, to address all of them during the live session, but we'll gladly follow up with the individuals who ask them directly. So if we don't get to it here, we'll get to it afterwards. But let me select just a couple. One of them, and we've touched on this, but the question was asked, how do you clearly distinguish what should be a study-specific KRI and what should be a QTL? Any specific thoughts on that? Any thoughts from any of the panelists on that one?
I'll throw one in there.
Go ahead, Steve. Go ahead. You go first.
Well, QTL, to be a QTL, we have to have a clear idea of what a threshold would be for the study is doing poorly at a study level. Oftentimes we have risks. We don't know how to set it for the study, but we know that if we bring it down to a KRI level and I'm looking for sites that are doing poorly, I mean, that's still going to help me, even though I don't know exactly what I'm looking for. So one thing I think is just if we're in a position where we have less information than we would need for a QTL, we could set it as a KRI.
Yep.
I fully agree with that, Steve.
Sorry, please go ahead, Marian.
Yeah, no, I just fully agree with that, right? I see KRIs much more operationally focused. It helps you drive and understand where and when you need to take an action, just at a day-to-day basis, at all different levels, and you don't need all the detailed information necessarily to do that. So it can be a little bit simple quite often from a setup point of view, and still add a lot of value from an operational follow-up. Yeah,
makes sense. Yeah. Joanna, were you going to add something?
I think it would be to think about also the impact if you decide on the QTL. you will need to report the QTL deviations in the clinical study report, right? So you really need this transparency with the study team from the very beginning. And also, like, I mean, this is maybe not the main differentiator, but the QTLs being reported, or the breaches being reported in the CSR, the signals really need to be meaningful, because we are going to support it, to provide them to or share with the authorities. So I think this is also giving us a little bit of differentiation here.
Yeah, no, that's a great point. It makes it sets the bar a little differently yeah, for QTLs. So let's see among the other questions. So here's an interesting. It seems there's at least a couple of questions coming from the audience about this, and one of them, I guess, there's an assertion here that will QTLs remain relevant given their apparent omission from the E6R3? Now, of course, E6R3 is in no way final yet, but I guess we're getting some indications from people that there's indications that there may be some change or removal of QTL terminology. Anyone want to take that?
I can take the first step, and then the others can chime in. So even if the QTL, at least the way we have approached it at Janssen, is not specifically called out as such. I think if you look at the ICH E6, 8, 9, so the estimate addendum, you look at all of these holistically together, then it still makes, if you want to apply with all these three linked together, it makes perfect sense to put them in place. And I think referring back to the first topics that we addressed during this call is the ethical component of it. That by itself, for me, would be a very good reason to continue with implementation of quality tolerance limits.
Makes a lot of sense, Marion. Anything to add, Joanna or Steve, on that one?
I mean, I couldn't agree more. I mean, basically, there is no different ask in the E6 R3 than it has been made in R2 or the recent revision of E8 and E9 as well upcoming. So basically, we are being asked at any stage to develop a holistic, like Mario mentioned, a systematic and prioritized risk-based approach to the clinical trial. And QTL, it doesn't matter, what kind of wording is going to survive after the public consultation in the upcoming revision to E6. I think that this still has tremendous value, and we should not just remove it from our centralized monitoring and RBQM approaches only because the wording has changed.
Yeah, makes a lot of sense, and I would have to say I agree with that too. I mean, like you're both saying, this is still early, and we don't know what the final R3 language is going to look like. But it sounds like, regardless, the expectation to be monitoring systemic types of risks and important risks quantitatively using ranges, whether you call it a QTL or not, is still going to be there. And then there's the ethical spirit behind it, as you mentioned, so it makes perfect sense. Let's see, I think we have about a minute or two left. I'm just going to, one more quick question, and then we'll wrap up the session. Do you adjust your threshold, I guess your QTL threshold or limit, during the study, or must they be defined before the study and not modified after? Steve, any thoughts? Can I pick on you for that one?
Yeah,
first?
Sure. We try not to, but we treat the QTL documentation as a living document. I believe we've done that once or twice when new information became available early on. The one thing we'd never do, though, is redefine the QTL if we breach it, and then retroactively make a breach go away. That would be completely unacceptable. But if we obtain new information that makes us adjust things, yes.
Excellent. And anything else to add, Joanna or Marion, on that one?
yeah, I fully agree with this. They are being defined upfront based on this expertise and historical data and not changed at any condition, because this is, yeah, basically it was cheating what our principles and ethical.
Yep, makes sense. Okay, Marion, anything to add there before we wrap up?
Perfectly in line with what the others have said, right? We rather not, but if you know that you made a mistake in your first, putting together, setting the threshold, then it makes sense to adjust that based on new knowledge, or things changed in the setup, might be a good reason to do so. Otherwise, nope.
Yep, makes sense. So thank you very much. We're going to have to wrap it up now, and like I said, questions that we didn't get to, we'll plan to follow up with you for any questions that you entered into it. So I want to really thank our panelists, our experts on QTLs for joining us today. Joanna, Marion, Steve, thank you so much for joining and sharing your insights. It was really a fascinating discussion today. Thanks for everyone in the audience for joining us, and thanks for all the questions. We really appreciate it. If you'd like to request a recording of today's session, please reach out to the email that you see on your screen, contact@cluepoints.com. And we also welcome you to join our next session of today, the panel RBQM Diversification, TSDV, TSDR, Dynamic Site Monitoring Strategies. You can seamlessly transition to that session by clicking on the Take Action tool in your screen to join. So hopefully we'll see you there, and enjoy the rest of the show. Take care, everyone. Have a good day.
Thanks, everyone. Bye, Steve.
Thank you.
Bye.
Thank
Bye,
you.
Steve.